Good. So good morning, everybody. It's fantastic to be back here. It's good to see old friends. We're here for the start of some of the important things in our lives. So this place has been one of those career-defining moments for me. So I'll talk a little bit about that. I should acknowledge some of my co-authors. I've underlined here 3 people who were with me here: Julia Condotti, Melissa Reynolds, and Maya O'Neill. They have told me I'm not allowed to add or say anything about their talks and steal their thunder, but I will nevertheless do something with that. I'm going to focus on what we have been achieving in the post-genomic era of eucalyptus, but first do a little bit of a recap of where we started. I think this is the birth. I wasn't at the 1999 meeting because I was still a student at the time. But this is what I remember. This is shortly after we had a meeting. This is my photo says Friday, 23rd of July, 2004. And at that meeting, there was a decision that we should sequence the genome of eucalyptus. Of course, the poplar genome was already in advance at that time. And you can see Jerry was there in the back, all young and fresh. And so this for me is kind of a career defining moment in terms of us making a commitment as a community to sequence the genome. Here we were still thinking about sequencing the globulus genome. Later on, of course, the granulus genome became the main target. And I have fond memories of that meeting here. You can see in the background we also decided to form the Eucalyptus Genomic Consortium, and it became Eucagen, the genome network, later on. We had people like Dario Guerapaglia with us. So Dario, unfortunately, cannot be here today. He told me last night, 12 o'clock via WhatsApp, to send his best regards. He is up and running, and I would say he's given me a couple of slides and some interesting developments to share with you. So Dario is definitely here in the spirit. And something else to remember about it, this is a moment that I will never forget, is that night we went out to Brad Potts. I don't know if it was your house. It was Brad's 50th birthday, and we had a massive big fire with an effigy at the top of Brad saying goodbye to his youth. I think a lot of us that are here were there that evening and remember. We only got to eat probably at about 12 o'clock. The guys who made the meat, I think they had a little bit too much to drink, so only the outer 1 or 2 centimeters of the meat was cooked, what I can remember. Some very fond memories of what we have done. And so back then already, we viewed this as a generational challenge. So thinking about eucalypts as probably one of the most important genera in terms of hardwood genera that's going to play a role in the future in terms of becoming sustainable on the planet for biomaterials and bioenergy. Fiber, of course, we should also think of other hardwood products. And that has pretty much defined what we did over the next 15 or 16 years. So fast forward, in 2014, the genome was sequenced. This is not the actual front page. We made this is the mock-up that the JGI did for us. It didn't get accepted. Some other less important water project got the front page. But we actually made a poster of this. We like that very much. Also, the companion paper, companion edition was the New Phytologist edition that came out shortly after. I think it was towards the end of the year, 2015. And many of the companion studies that did expert annotation of the eucalyptus genome, those studies were published there. So the genome paper itself, I had a quick look again last night to just remind myself what were the main findings. So, this is how I've heard the genome being summarized. There are many genes, and they are on a linear order. That's sort of the most accurate way to describe the genome. Dario recommended me that we should say this is a very high-quality assembly. From the start, the JGI did a fantastic job assembling that genome. It's one of the last major plant genomes sequenced with Sanger sequencing and a lot of back-end sequencing. And so it assembled into 11 pseudomolecules from the start, very long contiguous sequences. And it is still today, those of us that are using it, finding that it's a fantastic genome. It has been updated, but the quality, the second version of the assembly is actually very similar, just a couple of rearrangements. So it shows you how good it was from the start. At the time, it had the largest proportion of genes and tandem duplicates. Very interesting finding that many of these gene clusters were expanding at rates that we haven't seen in other plant genomes. It has this early lineage-specific genome multiplication event. About 95% of those genes have been lost over time. So many of the genes we work with, this is quite good for, these days we're doing, thinking of designing genome editing constructs. So many of the genes we're interested are in single copy, whereas we're also working in poplar. Many of the same genes there's 2 copies of those genes. There was evidence for complex genetic load, and some work has been done subsequently to show more detail in that, which underlies the inbreeding depression we see. And also, of course, there was high diversity for genes for secondary metabolites, so the terpene synthase and phenylpropanoids. And all of these are leads to work that has subsequently, so if we look at some of the highlights of what has subsequently been published, these were leads that were already provided by Eucalypt genome. And of course, many of us were interested in wood formation, so the lignocellulosic biosynthesis genes, and the genome gave us the opportunity to identify the bona fide genes involved in the different biosynthetic pathways. It's kind of fun. I went back to the Nature site last night and checked from the start. The genome paper was in the 99th percentile of about 190,000 tracked articles that are similar age, and it is still today in the 99th percentile, and it's in the 94th percentile of articles that were published in Nature itself, so 911 articles. So the genome is being highly cited still today. There are, if you look in Web of Science, there are 279 publications or citations, and the total sum of those are over 3,000. Also, there's some fun tools you these days can have on Web of Science, just looking at the authors that have published subsequently and cited the genome, so you can recognize many of your own names in here. Rebecca, Brad is on here as well, Dario, Jacqueline, where's Jacqueline, is right here somewhere, Jacqueline. Good to see you here, Jacqueline. Renee. So those are all of us that have made use of the genome subsequently. So there are some post-genomic highlights. So organizers asked me to highlight some of what I think are some of the, of course I'm very biased in this sense. I think one of the most important things for Eucalypt Genetics was the construction of the 60K SNP chip. So this was from sequencing of 240 genomes by Dario and his group. And this has really empowered for us a lot of the work that we are doing and that I will be talking about. This started already before the genome, but subsequently we've been using the genome SNP chip for that. So genomic selection really has been propelled forward. We have many studies with industry partners in progress. We might see some of that at this meeting as well. Then something that was impacted for us at the University of Victoria and many other people that are doing transcriptomics and systems biology, that reference genome has turned out to be very important for doing these high throughput multi-omic studies. And we've been able to press forward with this and do systems genetic studies, so expanding into large populations, doing hundreds of transcriptomes, and we would not have been able to do that without the genome sequence. I'm quite excited to see that we are getting closer. There's already, we'll hear about the sequencing of Corymbia, and there are also other genomes that are within the same family. So comparative genomics, I think, is going to take off very quickly. And lastly, I'm excited to hear, and this is Justin has given me some photos here, and Dean, that there are plans to sequence the diversity of Eucalypt species. So we'll hear more about that in this meeting, and I hear we're going to sequence in real time one of these genomes. Okay, so I mentioned the Illumina eucalyptus SNP chip that was made by Dario and in particular his bioinformatician, Ozanil Silva Jr. So this has been available, those of us that were involved early on, we were been paying $51 per tree, and it has 60,000 markers on the bead chip, Illumina Infinium bead chip. And the success of this is actually demonstrated by the number of samples that were within the first 3 or 4 years. Over 35,000 eucalypt trees have been genotyped using it by the diverse number of institutions worldwide. And now we are moving forward. So Dario has negotiated with Thermo Fisher, and they're moving into this 384 format. Many of you would know about this. This project is ongoing right now. Because we had such high demonstration of high usage, they were willing to bring the price down to $20 per tree. And for us, those of us that are working closely with breeding programs, in our calculations, of course, we want to have it even lower, but at $50, there are many species, many kinds of breeding that we do is actually not feasible. The benefit goes away, but at $20, it's sort of a game-changing price for many of the things we want to do. We actually want to see, of course, it go down to $10 per tree, but this is a very good start. So the objectives of that project, which is now ongoing, is to focus on the main commercially planted eucalypts. We also want to look at increasing the number of markers for some of the less represented species, like the temperates. There will be also at least 10,000 markers for Corymbia. So for those of there, it's been quite a bit of a demand for in many of our breeding programs, also in South Africa, moving into Corymbia species, sort of linked to climate change in some sites where Corymbia is now doing better than eucalyptus. And also something that will be of interest for those of you that are doing Eucalyptus genetics. We've included chloroplast DNA markers in the design. And this will also be done in a way to ensure maximum portability. So from the chips, so many of us, we already have thousands of profiles for the old chip. Want to make sure as many as possible of those markers. It looks like about 30,000 markers were informative across all of the studies. Many of us provided back the data that we produced with that chip to Dario, and they've been using that to pick the markers that should go onto the new chip. And so in summary, it's to really have a platform that's more economical and will be used wider, stimulate the adoption of eucalyptus SNP markers even wider than it has currently been. Last night, Dario sent me a message saying that actually there's been an important development because we have been able to immediately, so when we put out the word, 29,000 samples were pledged within the first month or two. So the company has been willing to actually make 2 of these. So the process of making these chips, there's actually a screening process where they first do a high density 650,000 marker chip. And now they have agreed to actually make 2 of these. So we will be screening a total of 1.3 million chips across 480 plus, so 960 samples that cover the diversity of eucalypts. And Dario says that I can tell people that the company has agreed to take the best 650,000 SNPs and to make that available as a commercial product as well. So there will be a SNP chip, we think just under $100 per sample, in which there will be a high, so in theory we should have a marker about every 1,000 base pairs. So for those of you interested in doing GWAS and higher density SNP marker analyses, that will be a product. We didn't foresee that, but it will be available. Thermo Fisher will provide it as a service. Of course, the production chip is going to be the 65,000 marker chip. So, okay, so I'm going to switch gears a little bit and talk about what we do at the University of Pretoria, some of the work that is being done in my research program. We started this around about the same time when the Genome Initiative started in 2003. As you can see, it's heavily supported by forestry companies, and so most of what I will be talking about is very applied, but we also do quite a bit of fundamental genetics. So our focus has been, of course, to extract as much value as possible out of the genome and move that into developing superior trees. These days, much of what we do in South Africa, and I think there's also in Australia, New Zealand, and others, there's now an international bioeconomy forum. And part of that forum, there's a forestry bioeconomy forum. They met in Canada recently. So, so going back to what we said in the beginning, going to a sustainable environment, so moving from a woody biomass through, if you're looking at all of the products that are already produced and some new products in the product line, so specialized biomaterials and green chemicals. So this is definitely the focus of what we are doing in our pulp and paper companies are rapidly moving towards these circular economies. And so, so if we look at the products that we will be pursuing, bioplastics, packaging materials are still, it's a big growth area still because we're all now ordering from Amazon. My son sent me a WhatsApp last night. I have to have something delivered from Amazon here before I go home. So all of that is packaging. Okay, to achieve this vision, we have to domesticate eucalypt. So this question of domestications, I don't know if Jerry will talk about that again. This is actually Ron Sedroff, my PhD supervisor, long ago said, we have to think about trees essentially as still undomesticated. I'm not quite sure that that's true. I think we've already gone some ways towards domesticating them as fiber crops. I think most of the sort of big changes may still have to happen at the wood chemistry level and other, so we start engineering the genomes, maybe that will be the kind of domestication that we will be doing. So, there are some visionaries looking forward to say, how are we going to go forward, going to Mars and many other areas. So Elon Musk said there are the three most disruptive technologies, and I wonder if people can guess what they are. There's a nice talk about the YouTube talk that you can download about this. He actually said the very first one is genetics. Genetics scale up. If we have control over the genome, we can do many things. And if you do it in microbes and in plants, the scale of what we can achieve is phenomenal. Artificial intelligence is going to be important because the amount of data will just be impossible for us to interpret. And of course, robotics. So much of this is going to be empowered. We know what the impact of robotics already has been in genomics and biology. And so we think we are moving rapidly in this. This is sort of the view of what we have of domesticating eucalyptus, doing things like genome editing, but also bringing information from spatial technologies, so looking at drone technologies and satellite technologies, so understanding the environment in which we are planting eucalypts. So this is pretty much the vision that we have in going forward. There's already, I'll say just a little bit about that, there's already a move towards high throughput gene sequences. We actually have one of my colleagues at the University of Victoria has a grant from the JGI who have now sequenced more than 200 transcription factor genes and promoters for wood formation tissues and pathways. And so we've already entered the synthetic era for eucalyptus genomics. Whether we're going to get to genome synthesis, I'm not so sure if it's even necessary. I think we will be able to achieve enough with the genomes that we have, and nature has already optimized them, of course. So my belief is that synthetic biology will become the dominant form of tree biotechnology very soon. So already we can just make synthetic promoters and design whole gene constructs, have it synthesized, and what a student, all of the months and months of cloning that students typically have to do can now be replaced by being way more productive and creative. Okay, so I just thought I'll show a quick slide about the program. So there are four academics who say more about what we do. So we're about 50 people. We're having a lot of fun at the University of Pretoria. So my own focus is more on population genomics, molecular breeding. So Stephen Hussey, a brilliant young, he was a Mandela Rhodes Scholar, and he's focusing on transcriptional control, and also he's leading the synthetic biology initiative at University of Victoria. Another brilliant young researcher, Esha Mizrahi, he's been working with me on resistance biology, and he's also starting a new focus on evolutionary biology of wood formation. And then Sanushka Naidu, she's one of the current Next Einstein Forum Fellows in Africa, and she's working on resilience to pests and pathogens. So working using some of the same technologies to look at, of course, there's a big problem in South Africa and worldwide with pests and pathogens moving in, and so we continually have to deal with that. So many of you would know this work. Much of what we've done in the past decade has been to capitalize on these populations that we've been able to make with our industry partners. So most of these are interspecific hybrid pedigrees. We now have we will soon have over 1,300 transcriptomes that we have. So you can see these are xylem transcriptomes that we have sampled. These are the 8-year-old trees, so we did it at 3 years and then sort of 8.5 years old. And so just using especially the SNP chip, we can rapidly make genetic markers, and then we can look at gene expression and then many different growth and wood property and bioenergy traits, some of those that we've done with Jerry and with NREL. And for me, again, whenever I get to this, I want to show this because for me it's one of the most beautiful things as a geneticist, the ability to track DNA segregation throughout. This is the 11 chromosomes of eucalyptus, and the power that we have to then do locus-specific association. Of course, I'm talking about QTL mapping. I mean, the other way to think of this is if we had to make these, we talked about genome synthesis. This is a form of genome synthesis. We have used segregation to make many different combinations. So these are almost the equivalent of hundreds of transgenic lines where we've mixed donor alleles. And this would be the foreground selection, but you have these alleles segregating in the background of almost all of the allele combinations available. Of course, these are all permissible, so there are many that we won't see. There are some trees in this population that died very early and that were runts. And so this has helped us to put together these gene networks, and we found throughout the genome these what we call hotspots, major regulatory regions that are controlling gene expression throughout the genome. And these are just transcription factor genes that we have been tracking. So these are major secondary cell wall transcription factor genes. So we've published this. I'm not going to say too much more about it, so you can read about it. There's a PNAS paper. We're now busy putting out a number of subsequent studies that are describing the outcomes of these. And so the good thing about the systems genetics approaches is that we can begin to link wood properties, complex wood properties, to groups of genes that are associated with them through multiple lines of evidence. So I don't think we've scratched the surface on this anymore. So we recently had Dan Jacobson from Oak Ridge with us as a visitor and plenary speaker. And I'm very excited to see the kind of things that he's doing in Poplar, and I think we will be pursuing that as well. So if you're interested in the amount in all of the data that we've produced, you can look at this at Plant Genie. The Plant Genie database now contains eucalyptus, Euc and all of the gene expression data is there. This is work done by Nanette Christie, a postdoc in our group. And she's developing this new tool called QTL Explorer that is making the transcriptome data available, but at population scale. So both for QTL, eQTL mapping, but also she's working with Nat Street at Umia Plant Science Center to bring in the GWAS kind of data. So EWAS studies, so SNPs associated to gene expression. That's similar to the big project, the 1,000 genome project that's been done at Oak Ridge as well. So we're making tools to make this available, and we hope we invite the community to make use of those. This will soon be launched publicly as well. And then out of that, of course, come the candidate genes that we can look at. And so only one of the ways we can do is through CRISPR-Cas technology. So we're doing both editing, but also the repression activation forms with the DCAS that's available. Okay. And then I think I mentioned already the synthetic biology. We think this is really the frontier of eucalyptus, is making the synthetic constructs and machinery, and Stephen Hussey is fast moving forward with that kind of research. So the last bit of my talk, I want to focus on what we are doing in terms of population genomics, more towards genetics, so what many of you here would be interested in. And we, focusing here on the high throughput market development and our future vision of how this will be applied in eucalyptus tree improvement. I've already mentioned much of what we're doing in terms of forward and reverse genetics. So most of this work is going to be more forward genetics that I will talk about here. But we also now have to add this component. So looking at trees on the landscape, understanding the interaction of the genome and phenome with the environment. So, so much of the work we're doing right now is population genomics, but we are also beginning to capitalize on the field trials that our industry partners have put out. And this is a very rich history of breeding. Some of the species, Eucalyptus grandis, has been in some form of breeding and improvement for almost 100 years in South Africa. So we have, I think, 5th and 6th and possibly the beginning of 7th generation material. In fact, it was a problem for us to do Eucalyptus grandis because we couldn't get our hands anymore on the 1st generation material. Those trials have all been removed already. Okay. And so back to the SNP chip. Most of this work that I will talk about now is we are doing with the eucalyptus, still with the 60K SNP chip. Here's a plug for Julia, my MSc student. So Julia is moving into these multi-parents. So most of the work we've done has so far been in single families, but we're looking at other designs. Many of you would know in maize, the Ed Buckler's nested association mapping approach. And so looking at these intersecting F1, intersecting half-SIP and full-SIP family designs. And the good thing is we realize that our industry partners have been making these crosses for a long time. That's how they breed F1 hybrids. And so there's a very deep trial series of these crosses that we can capitalize on. And Julia is one of the first students moving into that. This can now be done quite rapidly. She'll blame me to say the word rapid, but it's hard work still to make genetic maps. But it is, believe me, it's rapid. It's fast compared to when I was a student. And you can see we can make beautiful framework genetic maps. And you can look at this. She's going to give a short talk on Wednesday at 12:15. Now, these maps have allowed us to do QTL mapping of wood chemistry traits and a range of metabolite profiles as well. And they also allow us to begin to look across these families and begin to look at genome interactions. And so we can find regions, if we look across these half-SIP families, where there's strong selection for homospecific or heterospecific interactions. And I'll let Junia tell you a little bit more about that in her talk. I think this is a very important development because when our industry partners make these crosses, you oftentimes see complete incompatibility between grandis, and in this case, grandis and Urophila. It's not constant, so it's not that there's a particular genotype that's completely incompatible. So these are all specific combining features, and we can begin to deconstruct these at the genomic level. We've also done quite a bit of work with our tree breeding programs on genomic selection, and so we have 2 Eucalyptus grandis and Eucalyptus danii studies that are quite well advanced. We now have moved into validation populations, so we are busy looking at the next generation. These are sort of long-term projects. It's hard to get to the next generation. They have to grow up to some point where we can look at the phenotype. But we will have that soon and begin to report on that. One of the questions we have here is also looking at how we are deploying genotypes. We have a lot of information about individual genotypes, but sometimes we don't know exactly what we are breeding, propagating, deploying in the landscape. And these tools are very important in allowing us to look at genome composition, especially if you go into F1 and F2 hybrid breeding even. And so, we wanted to develop tools to breed and deploy these genotypes into the environment. And so we've started a project called the Eucalypt Genome Diversity Atlas Project. This is the student Stefan Henning who's busy with this, and Melissa Reynolds is sitting right in the top there, is leading this project in terms of managing the work. So we're focusing on the 4 main species grown in South Africa and also as hybrids, Euophylla, Dunnii, Grandis, and Nitens. And already just with you can expect this with the 60K SNP chips, 23,000 markers that give us very high, exquisite control ability to distinguish these genomes, and we can also see some cryptic hybrids that were provided to us as peer species, but there's definitely a hybrid of some sort. And then also drilling into the different provenances and regions. This is for Eucalyptus grandis, and Maya O'Neill is back there. We'll be telling you more about that. And similarly for Euophylla, just as an example, we can begin to look at the island level and see genetic structure there. What we wanted to do is there's a move towards F2 hybrids. So if we think of climate change, and so much of the trees that we plant are up the escarpment in South Africa, so there's an elevation range. And so some of the companies are moving into F2 grandis, Euophylla by grandis, for instance. And we want to know more about genome composition in these F2 hybrids. And so we've had a student who developed this pipeline. So you can use admixture mapping to do this. It's essentially a form of like chromosome painting, where you're looking at the number of alleles for one of the references. So with the 2 reference populations here, we're using the Genome Diversity Atlas, grandis and Euophylla, or grandis and Nitens. And I won't go into too much of the detail, but here's a PCA of genotypes, so we can distinguish very well Euophylla, grandis, and Nitens. And even some 3-way hybrids fall very nicely in between. And then we had this range. The company produced a range of, this is Mondi, a range of F2 and F1 genotypes. And we were able to, using this pipeline, very nicely paint the chromosomes back, and we could see the ancestry of different chromosome segments. So we can do that for F2 backcross and F2 intercross hybrids. And the neat thing is, if we look across the whole population, we can now get a profile across the 11 chromosomes. And for a breeding program, it's actually quite interesting if we, here's a case where this tree was provided to us as an F1, but we actually can see that this is most likely a backcross to grandis in this case. And even in the grandis backcross, there are some trees that are misidentified. So this is definitely not a backcross individual. This has Nitens. This is a grandis Nitens population, and we can identify cases where these trees are not actually what the company think it is. The good thing is, and where we're moving with this, is that we can now place these. The trials have been planted out across the escarpment, up and down. So there's a temperature gradient and also a pest and insect, pest and disease gradient. And so now we can begin to look at adaptation in these different environments and which of these trees are best suited there. So this is released via a database. So we made Bioplasm. It's a forestry genetics information system, and it has various kinds of information in it that you would recognize: trial data, species composition, provenance, genotypes, phenotypes, pedigrees. And this is to make the data available to our breeders and to allow them to very quickly apply things like genomic selection. So you'll see the poster by Melissa, poster 22, a little bit later. Then lastly, I just want to say, now we're moving towards understanding the genome interaction with the environment. We do have this kind of information where we can look at adapted genotypes. And the vision here is to leverage all of these G/E trials that the companies have put out in different environments, and then to begin to understand adaptation at the genome level. I call this applied landscape genomics, so leaving landscape genomics itself for natural populations. But the way we want to do it is more applied. And so Maya O'Neill will tell you a bit about the work we've now launched for Eucalyptus grandis, where we're looking firstly at the population genomics. She'll talk about the environmental association analysis that we've done. and then also the future plan for having large common gardens and G/E trials. And then really the vision for us is whether we, on the applied side, can do genomic models for genotype by site matching. I think it's still a big challenge, probably a big data exercise that has to be done. There's Maya doing a PhD. I won't go into detail here, but she'll talk to you about the environmental analysis. So there's great data here in Australia, site data that can be used for that, and we can begin to look into these. Is that the 5-minute harassment? Good. I'm done. I'm basically done. So this work, I think, is quite exciting. We're beginning to see the structure at structural level, the genomes, and associating that with different environmental variables. And so, of course, what we are interested in from the practical perspective from our breeding programs is understanding, after 100 years of breeding of eucalyptus, what has been selected, what has adapted in South Africa, and then can we relate that back to what's here, and even thinking about future environments. So there might be genotypes, and we know for sure there are genotypes and provenances that have never actually been brought into the country, and we might want to make use of that genetic variation going forward. So I believe we will be using this, the Oxford Nanopore MinION technology here at the meeting, so you will see that running. We're quite interested in moving over to the Promethean platform, which can do 10 times more, and some of our collaborators in the US have this available. And so, I think that is going to allow us to do the whole spectrum. So going from long-read sequencing to whole genome skimming. So this kind of low coverage skimming. So combining high resolution data with lower resolution data and progeny sets is where we think we would be going towards. And then also, of course, we can use the high density SNP chip and then the low density SNP chip, the 65K chip, to achieve that. And the future goal for us, I think, as in terms of our breeding program, and I'm hearing that also in Brazil and here in Australia, the vision has to be a haplotype-centric view of crop invasion. This is based on Mike Bevan's review recently. I think it is in Nature Genetics. Where should we go with crop improvement? And so what we really need to understand is haplotype diversity in these. And of course, eucalyptus, these are all inbred crop models, so we will we'll have to think of diplotypes and how they combine in these genomes. So to take home messages here, I think it's clear that Eucalypt Genetics has been massively empowered by applied genomic technologies. We are developing a genome diversity atlas, and I'm quite interested to hear what other people are planning to also map out the genome diversity of other species. We can resolve admixture and species ancestry now at subchromosomal level. That turns out to be quite a powerful technology for us to understand adaptation. We've embarked on a pilot landscape genomic study, and I am hoping to see that there are other people doing similar work in other eucalyptus species, so we can begin to compare tools and approaches. And maybe good news is there will soon be a cheaper SNP genotyping chip that will further advance genetics in eucalyptus. So that's the end of my talk. This is a bit of a vision that we have going forward for what can be done with tree biotechnology. So thinking of things like 3D printing and many other products. And with that, I'm going to thank you. And before I leave, I want to put a plug in for the IUFRO Tree Biotech meeting. I hope many of you are going to come to this. This is going to be in Raleigh in June of this year. And I'll leave you with some of the topics that will be discussed there. So we also have updated the program there. So some of these are new sessions that have not been there before. This is actually a proposal by Jerry, looking at the biotechnology for biomaterials and bioeconomy. So we're quite excited about some of the new topics that will be discussed. Thank you.
Thank you very much, Sander.
Yep.
We have time for some questions. Some burning questions? Yeah.
So, Sander, where do you think, so you're going were talking about that, the 650K screening chip at $100. Where does that compare to actually doing whole genome sequencing? You could get much more information out of that. And my quick over-the-head calculation, if you do the whole genome, if you do NovaSeq sequencing for less than $100, you get indels, you get tens of millions of variants.
Yeah.
Is it computationally too difficult to do whole genome sequencing?
No, I don't think so. I think it will eventually become whole genome sequencing. I had a bit of a 5-minute debate with Dario last night, and he's still making the same point. I made that same point, but the point is still in applied genetics, working with breeding programs. So you would typically do that. Let's say you have these design, you have 8x8 parents. You can very quickly genotype those, use the 65K production chip for the progeny. The big thing about this is turnaround time. So there's almost no bioinformatics because you get the SNP genotypes back, and the robustness of the data. So I think still for quite a while it's going to be an important approach, and so you can then rapidly also phase the segregation that you have in the progeny. But I agree with you. My view is also that eventually we'll move over to whole genome sequencing. But I'm guessing probably that SNP chip is going to still have a lifetime of about 5 to 7 years.
Thanks.
You showed some great information on the genomic side of things. Are you also looking at phenomics and new advances in phenomic technologies to complement the genomic program?
Yeah, so we don't grow any trees at University of Pretoria. We're quite dependent on what our industry partners are doing, but they are beginning to move into drone technology and remote sensing, working with other groups, so we've begun to collaborate with Justin. I'll be visiting with him afterwards. I do think we have to use phenomics. So we don't have that for instance, phenomics in the greenhouse. So we're planning to put up a facility like that. But it's still very expensive for us, so it's definitely where I think we should be going. It's just the infrastructure in the African context, it's going to be quite hard. But these G/E trials, and I believe Justin will talk about that, so having an international network of these trials, maybe there can be some technology sharing if we have drones that can measure them all in the same way and monitor them over many years. I think that's the way that we have to do it. We'll have to share resources maybe to make these happen. It will also be good to use the same technologies to look at trees in different environments.
You talk about all these white crosses that the breeders are making. Do you think that there's a lot of parallels with the natural white hybrids that probably exist here in Australia already, that these interspecific crosses that are releasing a lot of genetic diversity outside of Australia may be similar to what's going on here naturally that we haven't really discovered?
I think in some cases, yes, we have these hybrid zones and maybe going all the way from 100% of the one species and then integrate back to the other species. I think definitely, and those are have been selected over a long period of time. It will be very interesting to look at them. I mean, people have done this kind of work, for instance, in sunflower, and so with our hybrid species. I think it's something we can definitely look at, but also just remember the companies are also doing pure species, so that the pure species populations are still being taken forward, and those trials are also out there. So it's not just about the white hybrids. I think there's also much we can learn from the long-term breeding that they've done with, for instance, with Eucalyptus grandis.
I was just thinking about that in terms of that mixture mapping. Clearly, in a natural context with hybridization, we're getting those linkage blocks broken up into very small units. Do you think the forestry industry would be keen to look at that as a resource in terms of getting down to a much more genic level rather than those large blocks that are in the QCell on that?
Yeah, I think that's what I was showing at the end, moving to sort of a haplotype view. We need to understand, these blocks and what's the genetic diversity within these blocks. Of course, those blocks are going to be much smaller. They will resolve down to genes and maybe even half genes. We know in some cases the LD doesn't even extend to the other side of the gene. But still, we have to understand. But I mean, that's only true, when we look at big natural populations. When we look at breeding programs, where we have restricted number of parents, we are looking at much larger blocks of DNA. And so being able to sequence, sort of reset the recombinational clock by sequencing the parents and then looking at the progeny, we're talking about a different scale of doing things. But we are interested in tracking that back. So thinking of the 23andMe, those of you that have done 23andMe, the resolution there is quite exquisite. You can go down to small chromosome blocks and they can tell you that this piece of DNA is from an ancestor, in my case I was surprised, that lived in Scotland in the 1700s or something like that. So, once as the reference populations, the reference data is improved, and maybe that's something that the question I have for this community is, what's going to be the next project that we will do together as a community? Is it going to be a 10,000 genome project? Is it going to be a phenome project maybe? Or a G by E project? So we should think about how we can now again come together as a community to do the next big powerful science experiment in eucalyptus.
Thanks very much. I think that leads nicely into my question, which I don't have time to ask. So I think we'll save the remaining questions for the break. I think that would be really fantastic. And thank you very much, Professor Myberg, for a really fantastic talk.
Off to a great start. So considering the group that I'm talking to, I probably don't need to spend too much time on the introduction to eucalypts, but I thought I'd just put in a few slides just so everyone knows what a corymbia is. So eucalypt is the common term that encompasses the genera Eucalyptus, corymbia, and angophora, which covers about 900 different species of trees. They're very important for pulp and paper in the forestry industry, as they are grown in over 100 countries across 20 million hectares of land. Now, despite corymbia actually being classified as eucalyptus up until, I think, 1995, they're actually quite different from each other, having diverged more than 55 million years ago. And since that time, they've gone through some pretty significant changes in their genome size. So the estimated genome size for corymbia is roughly 400 megabases, as compared to eucalyptus, which is 640. So there's a considerable size difference there. So I'm sure others in the room, such as Brad, can tell you all the intricate differences between all the eucalypts. But the key that I tend to use in my head is that it's mainly the differences in the flower structure that allows you to distinguish them. So whether or not they have an operculum, so a cap that covers their flowers before they emerge, and what the structure of the flowers are once they emerge. So this is a corm structure, which is what corymbia is named for. So regardless of where the flowers emerge off of the main stalk, they all come to roughly the same plane, giving it a tabletop appearance for the flowers. So Eucalyptus and corymbia both have an operculum. Corymbia and angophora both have corum flower structure, and Eucalyptus and corymbia have an alternate leaf structure, whereas angophora has opposite leaves. So this is the species that I'm going to be talking about today. So this is a picture of Corymbia citridoria variegata. It's prized for timber in Queensland. It's grown for its nice straight timber. It produces a nice, beautiful hardwood it's very dense. It's great for the timber industry. Corymbia also has incredible growth rate. So I'm the one who took this picture here. So I'm about 6 feet tall. So these trees are roughly 8 feet tall here, and I was told that they were planted as seedlings about this height here. So in the course of 1 to 2 years, they've grown, gained about 7 feet of growth. So they're really quite incredible what they're capable of producing in just a short amount of time in terms of their biomass. Corymbias are also known for producing really wide hybrid crosses. So this is a Corymbia torreliana tree. It's grown up in the rainforest, and people plant it around their gardens because it makes a great shade tree, as you can see. What they found is that when you plant it down in Queensland, it tends to hybridize with the native forest of variegata, and it generates these hybrid trees that outgrow either parent by about 200 to 250%. And it seems to combine all of the desirable traits from both parents and goes into one tree. So this is what I did my PhD on, studying these trees for biofuel production purposes. In terms of the geographic distribution of Corymbia and Eucalyptus, so this slide is probably the one that I was most nervous to present, because in order to get both distributions on the same slide, I had to draw by hand. So I'm sure someone can point out that Eucalyptus probably grows a little bit further north than this, but just as a general rule, Eucalyptus tends to keep to the lower subtropical regions where it's much cooler, whereas Corymbia grows along the top end of Australia, places where it's much hotter and much more arid soils. So I wanted to point out Corymbia tessellaris here on this slide, just because this is its natural geographic range. It grows up from the top of Cooktown up to the bottom of Queensland or the top of New South Wales. This is the geographic range of roughly 2,000 kilometers. So across this range, there's a very wide difference in temperature ranges that are experienced, a lot of differences in rain, and wide range of pests and diseases that you can encounter from going from the tropics to the subtropics and through the rainforest. So there's a lot of bacterial and fungal pathogens that Corymbia needs to contend with. Those aren't very interesting to put on a slide, so I always like to show the giant wood moth, which lays a grub that's about the size of your thumb into eucalyptus wood. It loves to eat eucalyptus wood, and it's also the favorite food of the yellow-tailed black cockatoo. So these birds go hunting for these grubs. They rip apart saplings that have the grub in them, and then as soon as there's a storm or anything, the tree falls over. So this is what happens when you plant eucalypts outside of their natural range and they don't have any resistance to pests and disease like this. So the way that eucalypts tend to protect themselves against these things is to produce these complex terpenes, which are anti-herbivory phenolics that they pump into their tissues of leaves. The one that's from Eucalyptus citriodora smells great. It smells like lemons. It's great for the tree. It's not very good for me when I'm trying to extract DNA from it, because as soon as you release all of those oils into solution during a DNA extraction, they oxidize, turn brown, and destroy your DNA sample. So for these reasons, we wanted to sequence the genome of Corymbia. So with a partnership with the Joint Genome Institute, they generated about 120x coverage of Illumina data, 250 base pair paired end reads. We used that to generate an arachne assembly to get contigs and scaffolds. Back at UQ, I generated about 31x coverage of PacBio long read sequencing, which we then combined with the arachne assembly to fill gaps and stitch contigs together, make it more contiguous. And finally, we added long mate pair libraries that were generated at Hudson Alpha at about 40x coverage. We used that for additional scaffolding to put everything together as best we could. And then we combined those scaffolds with 3 DART-seq Corymbia genetic maps that were generated here at UTAS with our collaborators. And we built chromosomes using a program called AllMaps. So this is a table of the comparison of the Corymbia genome to Eucalyptus. So the number of bases that we got into chromosomes was 408 megabases. The total number of scaffolds in chromosomes is just over 4,000 pieces. Corymbia and Eucalyptus have the same number of chromosomes, both 11. Their GC content is effectively the same, at about 39%. But where they differ a lot is the amount of repetitive content that's in the genome. So there's almost a 10% difference in the amount of repetitive content between Eucalyptus and Corymbia. So across the board, Corymbia tends to have less in each of these different subclasses. But the largest difference that we saw was the long-term and repeat retrotransposons, where there was almost 8% difference of what was present in the Corymbia genome relative to Eucalyptus. Of course, it's not very interesting to describe a genome using a table, so this is what the genome looks like. There's a lot of information on this slide, so it's a good chance to take a drink of water. So starting from the outside band, so these are all of the 11 chromosomes of Corymbia with the different sizes of each along the outside. The next band in, so B, is all of the lines you see here are each of the genes that have been annotated into the genome, which have been overlaid with the number of genes per megabase density plot underneath it, so you can see where most of the genes are clustering. Below that is the average gene expression across our RNA-seq experiments with 5 different tissues. So because it's averaged across multiple tissues, you would expect that things that are switched on in one tissue would be off in another. So you would expect just a sort of a straight line across each of the chromosomes. So anywhere that you see large spikes such as these are genes that are constitutively turned on across all of the different tissue types. In from there, these are the positions of tandem gene arrays, which Xander touched on, that Eucalyptus has a large number of. So I'll talk a little bit more about these later, but these are their positions along the genome. The E band is the repetitive content in the genome. And finally, F is the positions of heterozygous SNPs within the genome. So the heterozygosity of Corymbia is roughly 1 to 1.2%, and these are the positions binned across the genome. So you can see that it's pretty consistent across the entire genome. It's very heterozygous, which makes sequencing a challenge. So to compare it to the Eucalyptus genome, I just wanted to see how close the nucleotide identity is compared to the 2 genomes. So this is the 11 Corymbia genomes compared to the 11 Eucalyptus genomes. And you can see that in terms of nucleotide identity, they're pretty similar. So the first 3 chromosomes, you can basically walk 1-to-1 between them in terms of their nucleotide content. And then from there, you get some small inversions, but all in all, they're pretty similar to each other. Of course, I'm a biologist, so I care about their genes and how synthetic the 2 genomes are. So the first thing that we looked at was the overall conservation of the gene synteny. So these are the order of Corymbia genes in Eucalyptus on chromosome 1. So anywhere that you see a synthetic block of genes, there is a little colored block, so if you notice there's a bit of fragmentation through this area. So this is due to small fragmented pieces that are gene rich, but there wasn't enough information to order and orient them based on the genetic maps that we have. So you see small little changes in their orientation. I'm more interested in the macro scale of these changes. So we ignore the small changes to treat, we look at large blocks of synteny and look for regions such as this where we see little inversions. So this is the piece of Eucalyptus that is inverted. Its orientation has been flipped around in Corymbia. We also look for things such as translocations. So a piece of the Corymbia genome that is at the end of Eucalyptus in chromosome 2 has been inserted into the middle. Chromosome 3 is effectively 1-to-1 synthetic between the 2 genomes. It comes out as one synthetic block. Chromosome 4 has one large inversion on the back end of the chromosome. Chromosome 5 is roughly synthetic as well between the 2 genomes. Chromosome 6 gets its own slide because it's my favorite. There were several rearrangements that has gone here. So the middle of Eucalyptus is now found on the beginning of the Corymbia genome for chromosome 6. So it has moved to the end like this. And then the back half of the genome has effectively broken and switched around like this. So what's interesting is that despite all of these rearrangements, all of the genes that belong to chromosome 6 are still there. There's only one interchromosomal translocation event, but effectively everything that I expect to find is right where it should be. It's just been moved around a little bit. So same thing, just finishing up the chromosomes. So chromosome 7 appears to have a little duplication event here. Chromosome 8, internal inversion. Chromosome 9, large inversion. Again, chromosome 10, large inversion. And chromosome 11, one last inversion. So this is a Circos plot between all of the synthetic blocks between Corymbia and Grandis. So just to describe what we're looking at here, anytime you find a large synthetic block in one genome, you draw a ribbon across to the other genome. So you can see across all of the different chromosomes, they're pretty syntenic with each other. These are the translocations that I was talking about. So a little piece of chromosome 6 from variegata can be found on chromosome 11 in this little block here in Eucalyptus grandis. So it's interesting to see that overall, over 50 million years, everything is still effectively where it should be. So the advantage of having another Eucalypt genome to look at now is that we can investigate the Eucalypt-specific whole genome duplication event. So a bit of background. The ancestor to all flowering plants went through a paleohexaplodization event, where it went through a large genome duplication event, which generated a lot of new gene pairs. From there, once all of the different orders have gone through and differentiated, some of them went through an additional whole genome duplication event. Poplar, I believe it was 50 million years ago that it went through its own specific genome duplication event. Arabidopsis went through 2. Soybean went through 2 as well. Eucalyptus, so the Natales group, it went through a whole genome duplication that is much older than all of the other groups. This occurred about 109 million years ago. And since Corymbia differentiated itself 55 million years ago from this, within this group, it should have evidence of this whole genome duplication as well. So just to explain what we're looking at here, so when a genome goes through a whole genome duplication, you generate a large number of syntenic genes that are no longer under functional constraint with each other, so they're free to accumulate mutations. So if we take a look at syntenic gene pairs and their synonymous mutation rate, you find that there is, this is the paleohexaplodization event that you can find using an outgroup such as grape. And if you go back earlier in time, you can see here that there is an additional peak. This is from the whole genome duplication that is specific to Eucalypts. It's a bit more difficult to see the Eucalypt-specific event because it's underneath the shoulder of this much larger peak, but it is there. So we can take a look at where these paralogous genes are in the genome. So you can see overall these are all of the connections between the whole genome duplication events. It's pretty similar between the two genomes. So that's the more ancient one. This is the more recent duplication. Yeah, so same pattern, but a few differences between the two. And because, again, I'm interested in genes, I want to know what's different between all these different sequence genomes. So if you take the R annotation that was provided by the JGI, we have 35,000 genes. We can cluster them along with other woody species that have been sequenced, such as poplar, willow, and an outgroup grape, and find what effectively distinguishes eucalypts, what genes come out as being shared in the eucalypt group that are not found in all of these other genomes, as well as what is specific to Corymbia and what is specific to Eucalyptus. So the first group, the Eucalypt shared gene clusters, these are the gene functions of what came out of the clustering. So I see a lot of zinc ion binding, ADP binding, lipid metabolism, and defense response. So zinc ion binding, if you have a zinc finger, you are able to interact with protein. Oh, I gotta speed up, I'm taking too long. You can interact with DNA. So Arabidopsis uses a lot of these genes to adapt to environmental responses, quickly turn on or turn off genes that are important. ADP binding is important for pathogen recognition. These resistance genes go through a conformational change, whether or not they have ADP or ATP bound, so it's important for recognizing pathogens. And defense response is fairly straightforward. Same things when you look at what is different between Corymbia and Eucalyptus. You see a lot of zinc ion binding, disease resistance, flowering genes, and heavy metal transport. And Corymbia was the easiest to describe because the genes that came out all were for defense response, stress response, terpene synthesis, and cell-to-cell signaling in some respects. So quickly, I want to touch on the tandem gene duplications. So as Xander said, 34% of the Eucalyptus genes were found in tandem gene arrays. It's really important for adaptive evolution. In Eucalyptus, these are typically associated with terpene synthesis and stress response, and they tend to be when you look within eucalyptus, it's a very dynamic range. That there are some genes that are really expanded out, some that are contracted back. So you can see what these tandem gene duplications look like. So you have a gene here that's in Corymbia that's duplicated 8 times in eucalyptus. And as you look through the genome, you can find more and more complex tandem arrays. So this is 2 genes in Corymbia that have been duplicated 8 times in eucalyptus. But these are the ones that I was most interested in. So where do we see an expansion of genes in Corymbia relative to eucalyptus? What are the function of these genes here? And unsurprisingly, we find, again, zinc ion binding, ATP binding, and genes that are related to terpene synthesis. So in summary, we have a new genomic resource for eucalypt taxonomy. Gene sensitivity between Corymbia and eucalyptus has been largely maintained with large chromosomal rearrangements. And we have gene family expansions in eucalypts with gene functions that are related to stress and disease resistance, cell signaling, environmental response, flowering, and terpene synthases. So what it looks like is happening is that it has these shared function genes between eucalypts, and then in order to diversify and expand out, it uses tandem gene duplication to generate new genes that are more similar to themselves than their counterpart in eucalyptus, and that's why they have clustered out on their own rather than existing in this shared gene function box. So this wouldn't have been possible without the Corymbia Genome Consortium. A lot of great work that has been put into this project. I'd like to touch quickly on some upcoming meetings. So this is a GGI-funded project. We have a GGI user meeting coming up in April, and CROPS conference at Hudson Alpha, talking about improving agriculture through genomics. And with that, I would be happy to take any questions.
Hi everyone, my name's Jack, or Jacob if you prefer, and I'll be talking about our analysis of the terpene synthase gene family in the recently published, well not published, but recently completed Corymbia genome, and contrasting it to previous work in eucalyptus. So terpenes and terpenoids are organic compounds present in plant essential oils, and they're fairly important. They mediate a lot of ecological interactions as primary and secondary metabolites, and they're also of high economic importance for pharmaceuticals and flavours and other kind of stuff. If you've eaten a eucalypt lolly, you've been sucking on terpenes. These are formed by terpene synthase enzymes, which catalyse the conversion of carbon precursors into the various terpene products. In this case, GPP is a carbon precursor, and various enzymes act on it to turn it into the different terpenes. These terpenes are divided up into different subfamilies based on the products they form, which range from TPSAs through to TPSHs, I believe. Luckily, there's been a lot of, since the publication of the Eucalyptus grandis genome, there was a lot of work done to look at the terpene synthase gene family by Kuhlheim and his associates. And so that's been really helpful in contrasting to the Corymbia ones. So since the terpenes are so ubiquitous across the plant world, they've been studied in various species. Of interest, though, is that in eucalyptus there's 113, which is the most expanded gene family across the entire plant world to this date. And you can see from this phylogeny that these are the Eucalyptus grandis TPSAs, and they're far expanded relative to everything else you can see there. So we were interested in finding out, is it as expanded in Corymbia? So since we have the lovely Corymbia reference genome to play with, I could get into that and have a look. So the Corymbia genome was automatically annotated, but I found that trying to pull apart some of the genes was a bit of a nightmare, so I went through and manually annotated it. So each of the terpene different subfamilies has an associated sequence with it. So the TPSAs have this sequence, TPSBs have this sequence, etc. And so I can blast that to the genome sequence. We find where the conserved sequence is located, and you can build out a gene along it based on its comparison to other genes and various gene features. And we also had some expression evidence to help out with those gene models. So in the end, we found out that there's a lot of terpene synthases in Corymbia as well. And they're all generally pretty well conserved in sequence, in structure at least. And compared to the other eucalypts that were studied before by Kuhlheim, it's pretty broadly conserved. So the raw numbers you see in the centre are how many genes are annotated. So globulus has 106, grandis 113, and we found 102 in Corymbia. And we also found that the proportional, the subfamily proportions were almost fairly similar as well. So you'd find that the TPSAs is about the same proportion in each of the different eucalypts, the TPSBs, etc., etc. If you compare it to something like Vitis, which has a much more expanded TPSG subfamily, or Ariza with the expanded E's, or the Arabidopsis with a much bigger proportion of A's. If you look at the raw numbers between Eucalyptus grandis and Corymbia, they're also fairly similar. There's sort of 1 or 2 differences throughout the whole lot. So it's fairly broadly conserved, which is good to know that across these different lineages of eucalyptus that the terpene synthases are still that expanded gene family. What we also found interesting, though, was that there's significant differences between the eucalypts, but they're at a finer scale, both within the subfamilies and in how they're arranged throughout the Corymbia genome. So there was a lot of, as you know from Adam's talk, the terpene synthases are arranged into tandem gene arrays, so tandem duplicates, so they have a tendency to expand and contract as you go through time. And so here's a phylogeny of the TPSC subfamily with the different eucalypts in different colours. You find that this TPSC gene is present in Eucalyptus grandis, globulus and Corymbia, whereas this one is only present in the 2 eucalypts. So either a gene has been gained in the Eucalyptus lineage or a gene has been lost in the Corymbia lineage. You can see a lot of this throughout the other subfamilies too. This is the TPSG subfamily. And if we look at this clade here, there's a whole lot of Eucalyptus grandis and globulus genes in orthologous pairs. But the Corymbia ones are floating down here by themselves and there's much fewer of them. So in this case, it's likely there was a common ancestral gene, but its evolutionary fate was different in both Corymbia and Eucalyptus, duplicating in different ways. We also found a lot of gene movement. So this is a representation of the 11 chromosomes of Eucalyptus grandis and Corymbia. And it's such a mess that we won't look at that too much. We'll look at just this bit. So this is chromosome 10 and 11. And so on chromosome 11, there are 9 TPSB2 genes in Eucalyptus grandis. But the most similar genes in Corymbia are found on chromosome 11, but also on chromosome 10. So there's been these genetically related genes now occupy different positions in the genome, so they've moved about since the divergence. You might think that this sort of stuff is due to the assembly. There might be sort of assembly errors and things shuffling around, but we have a second Corymbia genome floating in the background that we can use to verify some of these things. And of the 7, well, it looks like they're small, but of the 7 loci that have been translocated around, we found support for 3 of them in this second genome, which is sort of independent evidence that it's real, basically. So, to wrap it up, it's similarly expanded in the 3 eucalyptus species, which is good to know that the terpenes are likely important to evolution of the eucalypts. And yeah, that it's a wide gene family is mostly characteristic of the eucalypts, and it's been interesting to see if it's in Angophora as well. But the extensive variation within subfamilies and in the genomic arrangement was very interesting to see, and that's the sort of differences you can expect between trees that have been diverged for a certain amount of time, so for sort of 50 million years. And I'll wrap it up there.
So the last speaker for this session is Justin Borowitz from ANU, and he's going to talk to us about a project on 1,000 eucalyptus genomes. And I think this is a great idea, and I like the idea that it's actually possible to go ahead with something that probably not that many years ago would be considered mad.
I think that's exactly what I want to say today. Thanks very much. Let's go. So this idea was quite a few of you in the audience, a consortium that we sort of put together. I was part of the original Arabidopsis 1,000 genomes group, and when I came to Australia and got into eucalypts from some people here in the back, that's what I wanted to do, and everyone said that's crazy. And I think these numbers on the sizes have gone up of what's possible, and the prices have all gone down such that this is sort of a no-brainer. And I think the real challenge is, how do we do this as a community? How do we share the genetic diversity that we're all collecting? How do we share the data from provenance trials that are going in all around the world that Xander talked about? And that will go in, and that have gone in, since the trees are still alive, and often we can go back to satellite type phenotyping. So I'll talk a little bit more about the big picture tomorrow, but right now, and before we go out to take this photo and sample a tree, just to try to sequence a genome during this meeting. Let's hope it works. It's kind of a stunt to see that it really is possible now with these small sequencers. I just want to sort of put this in context, that what is a genome and where's the diversity. So we've spoken mostly about these reference genomes and assemblies. You know, the grandis was the first one, and now we've got Corymbia and several others, snow gum. So I think there's at least 10 sort of reference genomes ready, and we can maybe generate another couple even at this meeting. And I think, so 100 reference genomes should get at sort of the lineage sorting level, the really kind of diverged genomes. And then this top bubble, the sort of 1,000 ideas, is a bit more than the number of recognized species. And I think we're starting to acknowledge that there's a lot of introgressions and hybridization or trait segregation within these species. So I put quotes like species context. And that would become kind of the reference, because I think there's a lot of debate in what is this species and how is it related to the others. And so to have a reference set, such as the Currency Creek set that I'll show in a second, would be wonderful as a framework. And then the third sort of sphere there is this population or family or breeding type level. And there we're going to be at the sort of 10,000 samples, or doing it in batches of 100 plate format, and low coverage sometimes is enough there, especially if these are family lines and you have long-range linkage disequilibrium. So roughly that's, at this population-scaled independent maternal lines, the linkage disequilibrium breaks down so quickly, like we just heard in Poplar, that different SNPs in the same gene can be unlinked. And so we need pretty good coverage to type all those SNPs. The family level, you can get away with, which Xander was saying, $20 for arrays, or $10 kind of resequencing for both discovery and imputation of those whole genomes. And then for phylogenomics, for the deeper evolutionary history, we really like to see all that synteny and rearrangements and gene family pathway diversification. And so we need assemblies at that stage, and long reads are now available. And so what we'd like to do is set up, in probably Xander's community can sort of lead this, it's shared molecular protocols, sequence data, and informatics pipelines. And so we've already put a lot of this up in NCBI. It's kind of unifying those working in the forestry space with specific breeding families with those out in the landscape genomic space, or tapping into the wild genetic diversity. It's the same sort of approach, breeding for natural selection. The environments are shifting, soil types are extreme, and eucalypts can handle it. And so how do we deploy the genetic diversity in the right place at the right time for the future? This is already happening. I wish we had a newer slide, but this is at least a decade ago. I'm sure we're beyond the 20 million hectares. And these plantation forests are quite dense. So sort of you think about 20 billion trees, and the forests in Australia are not quite so dense. So even though we might have more hectares across the native range, the density is lower. So I often say that the number of stems outside of Australia is similar to those on Australia. But the genetic diversity is narrow when we go out. So there's a huge opportunity for eucalypts to take over, if you will. So one point here, and I know Mike, Chris, and Karsten will talk a bit more, but we have these sort of subgenera that are understood to be, reproductively isolated. But within that is a tremendous amount of genetic diversity and a lot of incomplete lineage sorting. So the gene trees that give us these traits, these morphological characters, the flower types that allow us to distinguish the varieties, are not necessarily following a whole genome-wide tree, right? And so this is a realization. A lot of plants have this, that the species trees and the gene trees are very uncoupled. And so we actually a lot of times want to know what the genetic basis for these morphological types are. And then we can predict their environments. And so this is something that Jason Bragg and I and others have been trying to find these gene sets, founder species that we could put back out into the landscape that will both be adapted for the local region and expected future conditions, but also be adaptable, be able to release some of that cryptic hidden variation that we're talking about when we make these wide hybrids that nature is also making. And how can that give us the ability to shift the means, but also play with the variance in these gene pools? And that might be a different set of genetic traits. So we could both select on adaptive alleles as well as background diversity, so that there's a lot of variation for these extreme conditions that we're recognizing. So what we're putting into NCBI is a Currency Creek reference set, which Dean Nicole is a real leader on this, and I'll show you a slide in a second of what last year we took, several labs went out and sampled the whole arboretum. And then Rose and others will talk about a few plates of samples that we've done within the Adnataria group. And BGI last year in their China National Gen Bank has really offered us subsidized prices for anything that we want to send. And David Bush and others have contributed, so we're starting to put that data up, as well as new samples that are coming. So I'll just sort of flip through. This was the team last year at Currency Creek, and we flew the drone up at about 100 meters. Ming in the back who took this drone shot. This is how tall one of the centurion tree would be. So some of those one day in the next few centuries will be up at 100 meters looking down at the rest of them. And the diversity coming out of this place in the back of the truck there is more than exists anywhere in the world, right? It's the hybridization that would probably happen is really beyond comparison. We need a lot more of these living collections that are releasing adaptive diversity all around the world. So let's catch up to Dean and put 100 of these out at least as provenance trials. Here's what we've done for Meliodora and soon for Albans and Cider-Loxolon. There's another project, big group on Jara that's underway. A new one from camel-based collection Jack Gillian did out in the Fenner School. And Karsten had proposed similar in Tereticornis. We have a huge amount of genetic diversity within and between species. And I just want to finish with this concept. This is the National Arboretum. In the back are these eucalyptus forests. And we have, I think, a huge diversity of habitats. And so really the genomics is just the baseline. This is just the beginning. What we want to do is put these into the ground at replicated common gardens. And this is happening for poplar, this is happening in maize. These huge G/E trials now are a limiting resource. And sharing this through some of these new apps, the Breeder API and Germinate, are the way the agriculture community is doing it. And if we follow along with the trees, we'll have multi-decadal phenotypes to add to it. So thanks for your attention. Let's go sample and do some sequencing.
Justin, I think that's a really exciting set of slides, so I'm not going to let you run away instantly. I'm going to take a question or two if there are for Justin. All right, you all want your photo taken. So one of the things that I have in this, I think it's really exciting, is the sort of longevity of these tree experiments and the short nature of funding environments. How do you sort of see marrying that? Because some of these things will last for decades. That's a persistent problem.
Yes, it is. You need like Department of Energy funding, or I'm part of a center of excellence that can sort of subsidize some of this, and that gives us 7 or more 10 years. I think in reality it's going to be this kind of community that everyone puts in a little bit here and there where we can. And block funding to universities that can sort of monitor these field trials in the long run. The seeds, stock centers, the data permanence. More sharing, I think, is the only solution. Yeah, aggregating small grants is the best approach.
All right. Are there any other questions?
Matt's got one in the back.
What are the barriers to sharing now?
Simply will, I think, and then this kind of legacy of we all are kind of working on our small independent grants. We need to do what we promised to do for this and get it out. And then the evolving databases, they aren't simple always. I mean, the NCBI just knocked our submissions back saying you didn't use their right barcoding framework. So it's in journals, they don't really enforce it. I think we're all tired at the end of the study. We just want to get our own thing done. So just try to go that extra step. It's no real barriers. Yeah, good question.